Hi, everybody. My name is Gary Laverty. I'm a faculty member in the Department of Biological Sciences at the University of Delaware. I have a small research lab with undergraduate students, and we work on the organism that you can see behind me, Tetrahymena thermophila, a single-cell eukaryotic organism, ciliate. And my interests are in mainly in chemical sensing, but also in the tolerance. This organism seems to have quite a good tolerance of hypoxia or low oxygen. So what we're presenting here is a kind of a case study where two undergraduate students, ended up intersecting, collaborating on a very specific issue that came up with my research in this organism. So I'll be introducing those students. They'll tell you a little bit about what they did and what their role was. And then my colleague, Dr. Carlton Cooper, will also be coming on and talking about a second part of our project here, which is his interest in cancer biology, how to get undergraduate students involved in cancer biology, but especially from the point of view of healthcare disparity or health disparities. So I'm going to share screen real quick so that I can introduce everybody. And you're seeing here the two students. Medhan is still an undergraduate student who is working with John Junk, and he's learned how to do a lot of incredible bioinformatic work and evolutionary phylogeny type work, structures, and so on. And Jessica, who you can see here, was a student in my lab. She graduated this past spring. She did a senior thesis in my lab, and her interest, as I said, was with hypoxia. And the idea was that we were interested in looking at a biochemical pathway called the glyoxylate cycle. And Jessica in particular worked with an enzyme called isocitrate lyase. So she'll introduce and talk a little bit about how she approached that project. And then Medhan will come on and talk about how his bioinformatics work sort of intersected with Jessica's work on the same enzyme. Dr. Cooper will come on and, as I mentioned, talk about his interest in cancer biology and healthcare disparities. And those are really the two sort of major ideas that we have that we're trying to develop. UD has a really good, or our department has a really good undergraduate research program, but it's a little bit difficult to navigate sometimes. Students need to, kind of on their own, need to find a mentor, and they need to sort of navigate a bit of a maze in order to get into a lab and to get research experience. Eventually, if they do get into a lab, the research experience is fantastic. But sometimes that happens with difficulty, or it happens late in their career instead of early in their career. So that's part of what we're trying to work on with our undergraduate research program. The other thing is, as I mentioned, is that this thing that happened, our case study, where two students happened to connect and work on the same enzyme, two students working in different labs in different areas, but found ways to collaborate. We realized, hey, that might be a really good thing to actually try to encourage or try to develop in our undergraduate research program. Get students talking to each other, collaborating with each other, forming networks, and so on. So I'm going to let Jessica start first and describe her research. Just
Okay, so as mentioned, my name is Jessica Rainey, and I completed a senior thesis with guidance from Dr. Laverty, and I used Tetrahymena as my model organism. So I wanted to give a little bit of background. Hypoxia is a term given for oxidative stress due to low oxygen concentrations. and hypoxic stress can actually cause a cell to change its gene expression. The mechanism for oxygen sensing in Tetrahymena hasn't been well studied, but hypoxia seems to cause Tetrahymena to undergo the glyoxylate cycle, and that's what's being shown here. So you can see around the circle here is the tricarboxylic acid cycle, and then the glyoxylate cycle refers to this bypass step here. The glyoxylate cycle has two enzymes that are involved, isocitrate lyase and malate synthase, and isocitrate lyase became the focus of my project. The glyoxylate cycle has been documented in some protists as well as bacteria, fungi, and plants. And then to induce hypoxic stress in the Tetrahymena, we used a Prox 110. So in conditions, I performed quantitative PCR to detect a change in the expression of isocitrate lyase, also known as ICL. I also performed biochemical assays to determine the change in ICL activity across the two conditions. By the end of the project, I had some promising, although not statistically significant, qPCR results that the Tetrahymena were upregulating ICL expression under these hypoxic stress conditions. And that's all I have.
Thank you, Jessica. Medahan, maybe you can pick it up and talk about how you got involved in working on the same enzyme and then what you were able to show.
Yeah. So my role was sort of when Dr. Laverty came to, I'm going to share my screen real quick. When Dr. Laverty and Dr. John Junk, they met up and Dr. Laverty was studying this enzyme, isocitrate lyase. And I also happened to have been looking for a project at that time. So it was convenient timing for all of us. And I got this opportunity to work on the enzyme. So our sort of perspective was, or our sort of goal was to look at the enzyme through different perspectives. And there are plenty of tools that are available for that. And here I have this poster that it can definitely be improved, but I feel like it's a good place to start just to showcase the tools that, that can be used. So a good place to start was by looking up a sequence for the enzyme. And with that, we would just use something like UniProt. You can just literally search Tetrahymena isocitrate lyase, and you would end up with a sequence like this. And then from there, you can take this sequence and put it into BLAST, and it'll pop out. I don't have the search here, but it'll pop out a bunch of sequences like it. And from there, once you have a ton of sequences, you can start making trees. And I just have a couple different trees here. And the tools, so for this, you can use plenty of different programs. I, for one, I like Genius. That's what I ended up using. And the different tools you end up using, they let you export the trees in different formats. And with one of those formats, or some of them, you can make a sort of a network like this here. And it's useful because, like, sort of in a classroom setting, everything seems perfect. But when you're making a tree with real data, and there's so many different ways you can make trees, you end up with a lot of different trees. So networks are a different way to sort of observe the connections between these different sequences. And then we can also use a tool like Genius, as I mentioned, to look to align all of the sequences and see important information about them. Like here, I have PI and hydrophobicity, and you can see if I were to zoom in here, you can see it shows you how it changes across the sequence. Now at the top, for example, we have what's called a sequence logo, and it shows you what sort of residue is conserved at that position throughout the many sequences you have. And then here I have, this is, I don't know how to pronounce his name, I'm sorry. I think Wang Qing, I'm sorry, I might be butchering that, but this is as if you were looking through a helix. Somebody wrote an R program for that, a package, and I was able to use that package to write my own script that would take in the input, which is some fragment of the sequences, and it would split those up into these two diagrams. It would just keep pumping them out for how many ever sequences you put in, or fragments. And it's got this useful sort of legend here, this key right here that can tell you different properties. And it's just a different way to look at the enzyme. And then here we have what might be one of my favorite tools, Chimera. So we've got 5 different enzymes here, and then there's, 4 different enzymes going down, but in each of these, one enzyme is the same, and that is the Tetrahymena enzyme, the Tetrahymena structure. And that was produced by this fire. You would just fill in this information, put your sequence here that you found from Uniprot, and it takes some time, but it generates a structure for you. And this is useful for when there is no known structure. This allows you to predict the structure for whatever sequence you have. And so I was able to use Chimera to load up the structure. This is the predicted structure for Tetrahymena. And on top of this, you can superimpose different structures. And that's what I have going on here for different organisms. And going across, you can split up, using Chimera, you can split up the protein into different, parts, the sheets or the coils. And then finally, there's something you can sort of extend into in Chimera called conserve that allows you to look at different conserved regions in sort of a 3-dimensional structure for that enzyme. And that's what I have going on here. And I think that's all I really have for you guys. But I think it's worth noting that everything I did here, it's just sort of just the tip of the iceberg with all of these different tools you can use. Like Chimera alone, this might look like a lot for some people, but this is literally just the tip of the iceberg for all of these programs that I was able to use, these different tools.
Okay.
That's all I have.
Thank you.
Thank you.
So just to come back then, the point, the reason we sort of went through this is that this is an example of something that kind of happened accidentally. 2 students doing research in very different with different skills and different backgrounds and in different places. But as it happened, we were able to find a single enzyme, in this case isocitrate lyase from Tetrahymena, and figure out ways for these 2 students to kind of intersect or have a collaboration of sorts. And the idea that we're thinking about for our undergraduate research program in biology is to, instead of letting this happen accidentally, is to try to actually encourage it. So what we're what we're trying to think about is ways to deliberately have students that are enrolled in our research program. And we have a course number at UD for undergraduate research in biology. And so the idea would be to take students that are enrolled in that for credit in that research program and try to bring them together in various ways. Try to form cohorts, maybe small groups, 4 or 5 students that, and that could either be random or it could be by trying to deliberately look for things that the students are doing, techniques that they're using, or interests that they might have, or something like that. So the whole point is to try to get students talking to each other, maybe with a facilitator, and perhaps finding ways that they could do more research or gain more experience by interacting with another student. So that's one part of what we're trying to do with our program in biology, and going to let Dr. Cooper maybe talk about some additional things we're interested in just to finish up. And do you want me to share that?
Yeah. Okay, so can you share that? Okay, while you're doing that, let me say my name is Carlton Rodney Cooper. I'm originally from King Street, South Carolina, so I'm a Southern boy, but here I am at the University of Delaware. My research is, I'm very interested in prostate cancer metastasis to bone, and more particularly, why is it so aggressive in African American? Now, having said that, as Dr. Laverty mentioned, yes, we're very interested in getting students to collaborate, because science is best practice when people bring unique skills and expertise to solve one particular problem. If you look at a lot of scientific papers, you rarely now do you see one author, you see a collection of authors. Well, this is also very important in terms of bringing on minority students, those who are minority classes. Now, what I'm showing here is just a model that I presented to a grant in an effort to address one major problem: the lack of resources to study racial health disparity in minority population. So what I've been doing since 2006 is I've been very active in educating the minority community here in Delaware, and then sometimes other states, about health disparity and misinformation, which is very rampant, such as the Tuskegee trial, and why that was dangerous, but why we have things in place to protect people from that ever occurring again. And the urgent need to have cells and tissues and a variety of tissue resources from groups that are affected most prominently by things like heart disease, diabetes, end-stage kidney disease, COVID-19, right? And my main interest is prostate cancer and breast cancer metastasis to the bone. So first thing we have to do is educate. So here you see I'm showing this, and these are just some community organizations here in the area. You could have some in your area. Explain to them the urgent need for minority participation in research, how that move research forward, not just for minorities, but for all people. Case in point, HeLa cells, Black woman, but it benefited everybody. So whatever we learn from studying African American cells or other certain racial groups, it don't just benefit that race, it benefits us all. And Henry L. Lack's story is a classic example. I think all of us are aware of that. What I'm trying to do in my particular area is isolate endothelial cells. What are endothelial cells? These are cells that make up your blood vessels. And you know, we need blood in order to transport nutrients, get rid of waste products and so forth. We need blood to live. Well, when these cells are malfunctioning, they can lead to a host of disease such as cardiovascular disease, diabetes. And my interest particularly is, does this also lead to more aggressive cancers that we see in African American men? African American men have the highest incidence of prostate cancer and the highest mortality of breast cancer, highest mortality of prostate cancer. African American women, they don't have the highest incidence of breast cancer, but they have the highest mortality. Okay, and the same can be said for things like cardiovascular disease, diabetes, hypertension, end-stage renal disease. I know that's of interest to Dr. Gary Laverty. So in this, we want to educate, aware, and stimulate and recruit more African American, and not just them, underrepresented minorities such as Native American, Latinos, to undergraduate research, where they will collaborate with a diverse group of people, including racially diverse, to solve a particular problem. Now, I'm not saying that it has to always be health disparity. There may be interest in other things, but still the underlying theme here is cooperation and collaboration. So now I'll turn it back over to Dr. Gary Laverty.
Right. So thank you, everybody. I think so. The point, what we're hoping to do is, and maybe hopefully people in this summer institute will have ideas or will be able to help us figure out good ways to do this. But we're really hoping to do two things. One is establish a more interactive undergraduate research program where students really have opportunities to talk to each other and maybe find common things to work on, as what happened accidentally with Jessica and Medahan. And also then to find ways to recruit and recruit underrepresented minorities and first-gen students, give them more opportunities, maybe even just giving them more information about how to get involved in undergraduate research